Structure of protein. Protein is composed of amino acid. On the basis of structure, 4 levels of proteins are found. That are primary structures, secondary structures, tertiary structures, and quaternary structures. Primary structures. And primary structures, the amino acids are found in the form of linear arrangement, and this linear arrangement is maintained by a covalent bond of disulfide bond. That is, this is a chain of amino acids, that is a polypeptide chain, and here the disulfide bond is formed in the form of covalent linkage. For example, insulin. Secondary structures, these are formed by the folding or coiling of areas between the polypeptide chains or within the proteins, and these are stabilized by hydrogen bonds. For example, alpha-helix and beta-pleated sheets. Alpha-helix, that is the structure of the alpha-helix, which is just like a telephone cord and is held in place by hydrogen bonds between every nitrogen-hydrogen group. There is nitrogen-hydrogen group and carbon-carboxy group, that is the oxygen group of the carboxy group. Between these two, a hydrogen bond is formed which keep them stabilized, and the typical alpha helix is about 11 amino acid long. So that is the structure of alpha helix. Another example of secondary structures is the beta-pleated sheets. In the beta-pleated sheet, it contains extended stretches of polypeptide chains. That is the polypeptide chain. There is another polypeptide chain, and there is another polypeptide chain with hydrogen bonds between the neighboring strands. That is the hydrogen bond between the oxygen of the carboxy group and the hydrogen group. So here the hydrogen bonds are present between the neighboring strands. Here in beta-pleated sheet, 2 sheets are found. That are parallel beta sheets and anti-parallel beta sheets. In parallel beta sheets, polypeptide strand runs in the same direction from N-terminus to the C-terminus. There is another strand, polypeptide chain, here also start from the N-terminus and end on the C-terminus. While in the anti-parallel beta sheets, the strand runs in opposite direction, that is from the C-terminus toward the N-terminus. Third, tertiary structures. These are formed as a result of non-covalent interactions among the amino acids, and they form 3-dimensional structures. For example, myoglobin. Number 4, that are quaternary structures. These are also formed as a result of non-covalent interactions by the binding of multiple polypeptide chains to make single larger proteins. Example of quaternary structure are hemoglobin that consists on 4 chains, 2 alpha and 2 beta chains. These are the primary, secondary and tertiary structures of proteins.